My name's Brooke Edmonds. I'm with Oregon State University Extension, and I'm organizing this webinar series. And I'm pleased to introduce our speaker. Dr. Ryan Contreras is our speaker this morning. He's an associate professor in the Department of Horticulture at Oregon State University. His research program focuses on woody nursery and landscape plants. Dr. Contreras also teaches courses on woody plant identification, growth and development of horticultural crops, and plant propagation. Research in his lab focuses on developing new plants that work well in production and serve the needs of growers and us gardeners. So welcome, Dr. Contreras, and we'll let him share his screen now. So just give him a few seconds to pull up his presentation.
Okay, super. Well, thanks a lot, Brooke, for the introduction. I appreciate the invitation. It's always good to talk to the gardeners because, as Brooke said, we do have a research program that's developing new cultivars for the nursery industry is who I principally work with, but obviously nurseries sell to independent garden centers and box stores, and we hope that they ultimately make it into homeowners' gardens. I'm gonna, Brooke is gonna do the moderating and stuff, but I do hope you throw questions out, and I'm happy to stop at any time. But otherwise, I'm just gonna get right into it. So today, let's see. What I hope to try to address today is to one, define what ploidy is, introduce if you're not familiar with that term, define that. Why would we use polyploids, or why would we induce polyploidy in our breeding program, and how we use that in breeding for disease resistance. And today I'm really going to talk about two examples from my program. But first, what is polyploidy and what is ploidy? Ploidy is simply the number of sets of chromosomes that an individual has, whether that's a plant or we humans happen to be diploid, as you see here. That means they have two sets of chromosomes. So a triploid is a plant that has 3 sets of chromosomes, and a tetraploid is a plant that has 4 sets of chromosomes. So we commonly think about most individuals being diploids. Plants are different than animals in this respect, as animals do not tolerate higher ploidy levels above diploid very often. So plants actually don't seem to be bothered very much by higher ploidy levels, and so I'll talk about in a minute that many of our crops are actually higher ploidy levels. So collectively, anything above the diploid level would be termed a polyploid. So when you hear that term, I've actually had people who have asked me about my genetic modification or GMO or GE breeding program, thinking that inducing polyploids is in some way genetic engineering. And I want to make sure that everyone understands it's actually quite the opposite. This is a very natural technique that is very common in evolution, and in no way genetic engineering. So it does happen commonly in evolution. So estimates have ranged over time from different scientists. But so going back to 1980, 70 to 80% of monocots have had some polyploidy event during their evolution. A similar estimate from Lewis around the same time in dicots. And then a more recent estimate was that 70% of angiosperms from Masterson. But the more that we've studied this and the more we begin using next-gen sequencing and other more modern tools, we find that actually all vascular plants have had some whole genome duplication or polyploidy event during their evolution. So you don't need to get bogged down in this tree here, this dendrogram. But if you look starting from the left of this is more ancient coming to more recent. So it goes all the way back to 450 million years ago. But if you look, there's an orange block that says that that's the ancestral seed plant whole genome duplication. So it goes all the way back. So that means that is the root of all seed plants. So there has been a whole genome duplication there. So polyploidy it impacts all plants. And then as you go through, there's been several that are confirmed by sequencing. The ancestral angiosperm whole genome duplication is in the purplish box there. And then there are several other gray ovals and diamonds that are additional whole genome duplications that are confirmed by whole genome sequencing in those crops. So it's common, it happens during evolution, and we just want to take advantage of it in our breeding program. So why? Why would we want to double the chromosomes of a plant? The first is sterility. So we, as horticulturists, we have definitely been guilty of introducing plants that have escaped cultivation and have either become naturalized or in some cases even invasive. So my breeding program, a large part of it is focused on breeding plants that don't escape cultivation. So we can induce sterility using polyploidy. You can increase the fruit or flower size. So these in polyploids are often larger than in their diploid progenitors. Altered growth form. This is one that I'm more and more excited about taking advantage of, and combining number 2 and 3 is really intriguing to me, where contrary to increasing the organ size, so you get larger flowers or fruit, you oftentimes will get a more compact plant. And so actually combining those things together, for instance, I'm going to show you a little bit of work on Vaccinium ovatum, our native box huckleberry or evergreen huckleberry. I think that could be a really big improvement in that plant to get more compact growth but larger flowers and fruit. In some cases, it can facilitate crossing. So sometimes during evolution, related species have evolved different ploidy levels. So if you double one species, that will facilitate crossing with the other species. And I'm going to go through and give quick examples of each of these 4. So here are some horticultural crops that many or most of you, probably all of you, are quite familiar with. And some citrus, the basis for seedlessness in some citrus is Triploidy. Bananas are all triploid, and I like showing this image that, you know, if you want an heirloom banana, there is one right there. So basically there are more seeds in a diploid banana than there is flesh to eat. So all bananas, including Cavendish, which is what we eat essentially, are all triploids. And seedless watermelons are also triploids. So again, it's a perfectly natural process that has occurred during evolution, and it's in many of our food crops that we eat. So how do you develop a triploid? So here you see a schematic of a plant that's represented as a tetraploid with 4 sets of chromosomes, and one that's a diploid with 2 sets. You simply cross them, and during the sexual hybridization or unification, you get 2 sets of chromosomes from the tetraploid, 1 set from the diploid, and it results in a plant that has 3 sets of chromosomes. And the basis for sterility is that during meiosis, when you have a reductional division that creates pollen or egg cells, you can't divide 3 equally. So you can't have a plant that has 1.5 sets of chromosomes in its pollen is going to be rendered sterile. That's, for all intents and purposes, that's the basis for it. You can't divide 3 equally by 2. So increasing flower size. This one's pretty self-explanatory. The plant on the right is a diploid. This happens to be an azalea dendron. It's a hybrid between an evergreen rhododendron and a deciduous azalea. And we wanted to do some more breeding work, but it was sterile. So we doubled it, and the plant on the left is a tetraploid, and it roughly doubled the size of the flower. It has an obvious ornamental improvement. Similarly, this is some more work from Vaccinium ovatum, or evergreen huckleberry. And on the far left is the diploid, so that's the wild type, unimproved form. And you can see the fruit volume is about 150 cubic millimeters. And we've, in our tetraploid, this is essentially an unselected population. So we haven't even selected among the highest or the largest fruit yet. But we've increased the volume to 200 cubic millimeters simply by doubling chromosome number. So it does hold a big potential for improving some of these small fruits, I think. Berberis thunbergii, so as part of our work, our lab and many others around the country are trying to work on sterility in Japanese barberry. And it often does result in larger leaves and thicker leaves that are darker in color. So larger leaves alone isn't necessarily a good trait, but these thicker leaves often have a thicker cuticle. They have deeper color, deeper pigment, and can be a little bit more drought stress tolerant. Similarly, here's on the right are diploid, a mermaple leaves. You can see the tetraploids on the left are darker green. You can't see, but they are considerably thicker as well, and they actually do have a thicker cuticle. So some really visible, obvious phenotypic results of inducing polyploid. Here's another example from some work I did down in University of Georgia. The plant on the right is Hibiscus acetosella Panama Red, which was selected by John Reuter as a better form of better red. It held up better down in the deep south. And but the comments from folks that visited the garden was that it got too large by August, mid-August. It was 8 feet tall and flopped over. So we actually developed a polyploid form of that, and you can see the plant on the left is much darker red, almost purple. The internodes are considerably shorter. It just provides an overall more compact plant. So that's what I was talking about, that adjusting the overall growth size. And then to facilitate crossing. So I mentioned evergreen huckleberry, and this is, I stole an image from the internet here, but Vaccinium ovatum is a diploid, while Vaccinium corymbosum, which is the cultivated blueberry, it is a tetraploid. We would like to take advantage of the amazing work that's been done by our predecessors on developing awesome blueberries. And so we would like Vaccinium ovatum. So that's why one of the reasons why we developed those octoploids, or tetraploids. So that's the next step. We will be making those crosses. And actually, previous work at North Carolina State has proven that this cross is able to be done at the tetraploid level. All right, so I'm going to give a couple examples of some work in polyploidy we've done in my lab. So if you're familiar with Cotoneaster, this is Coral Beauty, and this is right here in Corvallis, where we basically had no rain from somewhere around July 4th through, depending on the year, September. And this is how I often see Cotoneaster Coral Beauty used. It looks like green poured concrete. It's sheared. I mean, it looks like they come by weekly to shear this thing. So this is oftentimes how it's used. There's no irrigation in this parking lot, and it's tough. It does what it's supposed to do, I suppose. It's not my style, but the humble Cotoneaster can be an aristocrat with some nurturing. So here are two of my favorite uses of Cotoneaster that I've seen. The one on the left is Bouchard Gardens, which is up in Victoria. And on this hillside alone, there are probably 8 or 10 species of Cotoneaster. On the right is in Lyon, France. I had the pleasure of being over there, and just really softens this large concrete wall. That was Cotoneaster Dameri going all the way into this villa right here. So really quite lovely. Cotoneaster, a lot of people ask me why Cotoneaster. Well, one, it works in those parking lots. It's a very, very tough plant. Once it's established, it's super drought tolerant and provides multi-season interest. And what a lot of people don't know is there are actually more than 400 species of Cotoneaster, from tiny, minuscule, millimeter-long leaves up to basically small trees. So, and there's lots of different ornamental traits that folks haven't really tapped into. And there's just been a lack of breeding. This is also at Bouchard Gardens. I love showing this. This is, I call it the feature plant of the whole garden. Of course it's not, but this Cotoneaster Dameri is actually situated right in front of the lovely fountains here at Bouchard. Again, lots of different uses for Cotoneaster. It grows essentially right out of rock. There's very little soil here, very drought tolerant. The one on the right is a Cotoneaster apiculatus that's grafted on its standard, and it's just growing out in a median outside of Beaverton, Oregon. But this is without shearing those Cotoneaster Coral Beauty to within an inch of their life. This is what Coral Beauty does. This is in the parking lot at Reeser Stadium here on campus. I'm not bashing on our landscapes. They do a great job with what they have, but this is Coral Beauty is not a very good plant. Overall, it's just it's fairly rank in growth. It's very vigorous, produces lots of flowers and fruit, but this is a pretty bad form. And furthermore, I'll show you some data in a little bit. It's also susceptible to fire blight. So There's been very little selection for improved habit, improved form in many Cotoneasters, and also fire blight. So this is the typical characteristic shepherd's crook that you see in fire blight. You can see the young shoots get infected, and it gets that shepherd's crook that's so characteristic to this pathogen. So, okay, we wanted to breed for disease resistance. How do you get disease-resistant plants? Well, it's not magic. You have to find a source of resistance to use as a parent. And what we do, working with our colleague Virginia Stockwell, who's an expert in fire blight, she helps us mix up some inoculum of fire blight. So we take scissors and dip it right into this inoculum, and then we have vigorously growing plants in a greenhouse, and we go bisect the two youngest leaves on a very fast-growing plant. And that's what the technician and graduate student were doing here. They had inoculated, and then weekly we go in and measure the lesion length. And that basically gives us a relative measure of how susceptible or resistant a plant is. So when we started our work on Cotoneaster, we had 52 species. We put it in a greenhouse. We did this trial. We actually did it over 2 different years to get a gauge of true resistance of these plants. So then at the end, luckily, we did see many plants that were susceptible. The plant on the left, you can see there's the arrow, and there's also a little piece of tape there. That is the inoculation site. You can see that plant on the left. Basically, as soon as we inoculated it, it died within about 2 weeks. So, and you can see it died all the way to the ground. The plant on the right is a good source of resistance. This is Cotoneaster splendens. And at the site of inoculation, the leaf that we inoculated, or the 2 leaves that we inoculated, they themselves didn't even die. There was no sign of any infection at all. It just kept growing vigorously. So that's a good source of resistance. So, okay, so how does ploidy then come into play in breeding for disease resistance in Cotoneaster? Well, in Cotoneaster, there's actually a pretty broad natural ploidy series. About 70% of these plants, of the species, are tetraploids. About 15 are triploids, and only 10% are diploids. And the polyploids, according to the literature, are obligate apomix. Okay, and I'll discuss that in just a moment. But the caveat, we've developed some synthetic triploids that I'll talk about in a little bit. But lucky for us as breeders, apomixis is not good for breeding, but the diploids are sexual. So we have to identify diploid species that we can use as female parents. So I mentioned apomixis. Apomixis is simply the production of asexual seed. Right, so it is a factor here on the West Coast, particularly in southern Oregon down into California, but even in the Willamette Valley. Cotoneasters are a bit weedy. We see them out of cultivation, and apomixis can contribute to weediness because there's no need for a pollinator or a pollinizer. The plants simply they flower and produce seed regardless of environmental conditions for the most part. So freezes have little impact on fruit set and seed set, and yeah, so they tend to be very fecund and very prolific, and so that's all of the tetraploids in the genus. So to develop sterility combined with disease resistance, we're actually taking advantage of this natural ploidy series. So the diploids are sexual, so we use those as the female parent. The tetraploids are apomictic. So they don't work being a female parent, but we can use them very effectively because they have fertile pollen. We can use them as the pollen parent. And again, when you cross a diploid with a tetraploid, the result is a triploid. So what we did, we used Coral Beauty. It's a susceptible diploid. We use it as the female. And we crossed it with Cotoneaster splendens, which was a resistant tetraploid, Divericatus, which is a susceptible tetraploid, and Simpsonii, which is a resistant tetraploid. And the reason why I put in the susceptible tetraploid is we're also, in addition to developing new plants, we're also interested in learning more about the science. So in this case, we wanted to learn about how is disease resistance transmitted, or how is it inherited in Cotoneaster. So just to give you a graphic, this is on the left is Cotoneaster Coral Beauty. It's a susceptible diploid. We crossed it with a resistant tetraploid, Cotoneaster splendens, and there is our first little seedling, and that's our cute little guy. I'll show you that full-size plant here shortly. So here's actually the first graduate student with my program, Joseph Rothluner. Here he's making crosses on Coral Beauty. This young man, I think at the time, was probably 23, maybe 24, and the flowers are still small enough that he needed to use the Optivisor to make sure that he got all of the anthers removed. So you can see, if you look carefully along the stem close to his head, you can see there are pistils sticking up where he's gone in and removed the petals and removed all the anthers. And he'll then go through and apply pollen from the pollen parent that we're using. So there he is, just getting in there pretty close. You can actually see he's in the process of removing the petals, and there's an exposed pistil just to this side of that flower. So as part of the process, we do the emasculations, we make those pollinations, and then luckily in this case we got some seedlings. So here's the 3 seedlings that I'm going to show you today. The next step in the process is to see if those seedlings have resistance. And we're always trying to do multiple types of evaluations as we're going through the breeding process. So in the case of Cotoneasters, if a plant doesn't have disease resistance, we're not super keen on keeping it in the program, and it's probably going to get thrown out. In all of our breeding programs, if you can't make more of them, so if it doesn't propagate well, it's going to be thrown out, because the most amazing plant in the world is not worth anything to you as gardeners or to nurseries if you can't propagate it to make more. So it kind of works out conveniently that the next step in the process is we propagate it to make more cuttings and more liners that you see here. And that facilitates further disease testing. So the next step here, this is Dr. Stockwell is on the left here, and then some other folks from my lab. And so we had propagated these plants. They potted up into quart-sized containers, and they're growing vigorously in the greenhouse. And now we're about to re-inoculate. And so I am an academic. I can't get through a presentation without showing you some data. And so the arrows starting from the right, moving left, that's Coral Beauty. And you can see that it has a mean, in this study, it had a mean shoot necrosis. So that's what we use. When we say it's 11% disease susceptible, it means that the site where we inoculated it, 11% of the shoot is infected. So if it has a 10 centimeter shoot, it would mean that the disease region, or the disease necrosis was at 1.1 centimeters long. So that's how we express our resistance or susceptibility. So that's the industry standard. And then moving from right to left, the next one had 1%, and then 0 and 0. So those are the left 3 are the 3 cultivars I'm going to show you today from my breeding program. So we're okay with 1 or 0. That's pretty good, considering that the industry standard has a mean of 11%. So here are the second plant from the far left is Coral Beauty, and then my 3 cultivars are the next 3. So the first one I'm going to show you, this is not the cultivar name, but we affectionately called it Muffin Top because it's so tiny, and this is what it looks like. It does look like a little muffin. It just overflows the size of the container. This is actually a 7-gallon plant. So it seems very small, petite, dwarf, but it's actually quite vigorous in container production. It roots very well. Again, going back to the previous, or the data slide, it has 0% shoot infection. It roots very well. It looks really great in container production. It actually produces, so this is a 3-node cutting was stuck, and you can see the liner that it produces was actually a branch liner, very filled in the root zone. You can get lots of cuttings. So my research assistant, Tyler Hoskins, got 118 cuttings off of a 1-gallon container. Those were small cuttings, but nevertheless, it's very prolific in its production. Again, it looks good early in production, but here's what you all want to see, right? Does it look good in a landscape? And this is my own home landscape. Never mind the weeds and all the other terrible plants that I have here, but I'm really excited about this thing. It doesn't flower as prolifically as Coral Beauty, but I think it's an acceptable rate of flowering, and it produces a decent little fruit crop. But again, not as prolific as Coral Beauty, but this is, you basically never have to prune it. You never have to touch it. All right, here's the next one. This is the most unique hybrid that's in my Cotoneaster breeding program so far. Here it is out in our plots where we're taking a look at it. You can see it's starting to develop some fall color. Here it is in container production about a week later. Some nice yellows and oranges, still got some green in there. And this plant will actually go all the way, it's fully deciduous. So this is the only fully deciduous Cotoneaster so far that we're planning to release. It does well in production. Here's our friend and collaborator Sam Huang up at Monrovia. And again, this is a really, really dense, prolific rooting cultivar. Pink flowered. Does anybody have any pink flower Cotoneaster in your yard? Probably not. They're very uncommon in cultivation. So we are pretty excited about this pink flowering cultivar. Here it is in my home landscape. And it flowers prolifically and over an extended period of time, whereas most Cotoneasters sort of have a big pow, white flowers over a shorter period of time. This is going to spit out these pink flowers, not at an insignificant amount, but over a much longer period of time. So it actually has pink flowers on it for several weeks. So, and so far, I'm not making the claim that it's sterile, but it has virtually no fruit in my observations. This is in contrast to the naturally occurring triploids that are apomictic, and so they produce asexual seed that's clonal to the mother and in pretty good numbers. So in this case, there seems to be no evidence of apomixis, and I will make the assertion that at a minimum, we have greatly reduced the fecundity of this plant. So I'm not saying it's sterile, but I would put my name out there that it's not going to escape cultivation. So I'll leave it at that. So the third is actually somebody who visited my program said you should just call this Coral Beauty Improved, because it really fits the same landscape niche as Coral Beauty, and actually the same production niche. If you can grow Coral Beauty, you can grow this. It grows exactly the same, except for it's just much more uniform in its branching here. So these plants are unpruned, and you can see they are quite uniform. The plants on the left are my cultivar I just showed you. The plants on the right are Coral Beauty, and they're the same age. You can see it less rank in its growth, and it is less vigorous. Here it is in my home landscape. This thing is a very prolific in flower. I think it flowers as much as Coral Beauty, but its fruit load is not as heavy. So if fruit is your main determining factor in growing a Cotoneaster, this may not be the plant for you, but I'm a pretty big fan. And again, actually I think this one had 1% shoot infection. So it's going to be low, but not none so far. So here's just a summary slide. Coral Beauty is the plant on the far right. Then you have Muffin Top, the white flowering, and the pink flowering. So I think compared to the industry standard, we have one sterile triploid and two, they are diploids, but they have much improved disease resistance compared to Coral Beauty. So I think they're improvements. And just again, I mentioned the diversity of Cotoneaster. This is just a little snapshot of the pretty astounding diversity in this genus. And you know, some of our crops, like we've been breeding Ribes sanguinea and flowering currant for a little bit, and after our next releases, we're pretty much done. But I think Cotoneaster is a genus that is just going to keep going and going, and I think I'll probably be breeding Cotoneaster for the rest of my career. Here's how the process works. I'll give you a summary here. So we make crosses between parents based on data that we had from our disease and from our ploidy analysis. We then confirm the hybridity using flow cytometry, which if there's time at the end, I can talk briefly about, but it essentially determines what the ploidy level is. We then propagate it, we inoculate it, And if it makes it through those rounds, we'll then conduct field trials, which you see in the bottom left. That's a bunch of nursery folks that are visiting. But anyone is welcome to visit. Brooke has actually visited our field day and even shot some video of my ugly mug. And then if it still looks good in the field trials, we'll then conduct material transfer agreements with nurseries and release it to the general public. But at any point, if it fails, we take it back to the controlled crosses. So if it doesn't propagate well, we'll try to make controlled crosses with something else and go back. Or if the disease resistance isn't there, we'll go back to square one. So I'm going to make a hard left here. So I'm sure basically anywhere you are in the temperate United States, you have seen or encountered Prunus larisoraceus and possibly Prunus lusitanica, this common cherry laurel and Portuguese cherry laurel. So the problem, well, there's a number of problems with both these plants, but our breeding goals are to combine shot hole disease resistance and sterility. So this is, whenever I talk about our breeding program and I mention cherry laurel, this is the program or this is the project that growers get most excited about. Because if you're growing cherry laurels, this is what they often look like. This is not, I didn't stage this, I didn't actually shoot these plants. This is just what they look like under overhead irrigation. It's a tough pathogen to get around, to deal with in production because it's actually bacterial, fungal, or both. So it's not like you can really do preventative copper treatment, for instance, because then it can be the fungus. So anyway, it's resistance would be huge in this crop. And there's no apparent resistance that we can find in Prunus larisoraceus that you see here. Prunus lusitanica, on the other hand, does not seem to be impacted, and we see very little signs or symptoms on Prunus lusitanica under overhead irrigation or in landscapes. But what you do see are these insane fruit crops on this. So this used to be right outside of my building, and you can see the sidewalk is completely covered with the fruit. You track it into the building, birds eat it, and they drop it on cars. Birds also eat it, and they distribute it out into the landscape. And so this was, again, not planted, but this is a really impressive crop of Prunus lusitanica coming up here on campus about 5 years ago. So how do we want to address the issues of disease resistance and sterility in these two species? We want to develop a mule. So if you're familiar, a mule is a hybrid between a horse and a donkey, right? And there may be some mule hybrids out there, but I'm not sure. Basically, it's a breeding dead end. Luckily, mules are quite useful, and that's what we're hoping to do. So we started about crossing Prunus lusitanica, which you see here is the typical inflorescence during spring of Portuguese cherry laurel. And we emasculated the flowers as you see here. So we're removing all the petals and all of the anthers. Use pollen from different genotypes of Prunus larisoraceus to pollinate these plants. And just showing you, this was a serious team effort. Here's 3 folks up in a tree, and we're bagging the inflorescences to make sure that pollinators don't spread more pollen, and that we're only having pollen from our plant of interest land on the stigma. Here was an undergraduate at the time. She was very intent on her work. I just, anyway, I made a grand total of maybe 0 crosses in these 5 years that we were doing this work. So we really had great folks working with us. So we got super excited. We had this seed set over 3 years. We had a pretty decent seed set, low, but not unexpected in a wide cross like this. Over the years, we had we tried about 10,000 crosses, a little over 10,000 crosses, and we attempted, as you see here, this is Justin Schulze. He was a master student with my program, and tried ovule culture to rescue hybrids because it is a wide hybrid and it's expected to be hard. So you can't just make a cross, collect seed, sow it. So we tried fairly hard to get this. In 2015, this is in 2015 that Justin is holding here. we put about 100 seeds in culture, and we had 10 that we saw germinate to some degree, but alas, we had 0 hybrids. So for those of you keeping score at home, that's 10,000 crosses and 0 hybrids. That's not a good return on investment. One of the problems is that Prunus loriceraceus is a weirdo, and it has a very high ploidy level. It's a 22x. So I mentioned diploids, triploids, and tetraploids. This is a rarity among plants. It's not unheard of, but it is certainly a rarity to have such a high ploidy level. So Prunus loriceraceus has 176 chromosomes. Prunus lusitanica on the right is an octoploid, so it has 8 sets or 64 chromosomes. So in addition to just being different species, they also have vastly different chromosome numbers. So to facilitate that, we have double the chromosomes of Prunus lusitanica. So we now have a 16x group of plants. And I put these side by side. The octoploid is on the left. The 16x is on the right. And you can see that they are a little more compact. They look like they're a little bit slower growing. Okay, another data slide. I can't help it. So you can see that the height is actually reduced from about 25 centimeters at the time of observation to about 16 centimeters. The internode length is shorter, so that means the leaves are all more compact on the stem. And the leaves are thicker in the 16x plant. So, did it work? Do we now have hybrids between these? We don't really know. So we don't know if it's going to facilitate the cross because, like many induced polyploids, they're terribly slow to flower. So it's yet to be determined. But in theory, if we are successful in developing a cross between the 22x Prunus loriceraceus and the 16x Prunus lusitanica, we should have at least some plants that have resistance. And that would be brought in by the lusitanica parent. And we should have sterile hybrids. So in addition, like I said, it's a mule, so a wide hybrid between 2 related species. It's also going to be a 19x plant, which is an odd ploidy level that you can't divide equally by 2. So you can't have 9.5 sets of chromosomes and expect to be fertile. So what I said here, what's less sure is that will we get a plant that is worth growing? So getting something that is a hybrid that's sterile, that maybe has some resistance, that would be awesome. But it also has to look good. You guys have to want to grow it, and it has to be able to be propagated and work in production. So that is the main part of my presentation. I certainly have some more material if there's time and interest, but I hope that that was a brief look and you learned something about how my program is using ploidy to breed for disease resistance. And I mean, I give every presentation has a picture of my family. So it's my wife, Megan, and my son, Cooper, and my daughter, Edie. So thanks so much for your time and attention. And I will open it up for questions. If there are none, I will go on to the last part. But
so it looks like Lori has a question. She would like to know what the ramifications of ploidy are on pollinators and pollinator habit. And I don't know if that's necessarily something that you study in your research programs, but maybe you're familiar with that aspect.
Yeah, it's something that I can't answer. So I am interested in it. And so, for instance, we're doing some work and going to try to make some observations on the butterfly bush cultivars that are allowed to be grown in Oregon now. And some of those vary in ploidy level. So, it's hard to say. One, if it produces if it's greatly reduced in pollen load, that may be less attractive to pollinators. And likewise, if it produces a great reduction in nectar, obviously those things would be reduced. But for the plants that we're specifically seeking to reduce the fertility of those plants, I actually think it's a good thing to potentially reduce the pollinator attractiveness to, for instance, Prunus loriceraceus. I think we don't want our bees to be visiting common cherry laurel and taking attention away from other plants that we'd rather have them pollinating. Now on the Vaccinium ovatum, obviously we have to have them pollinated. So I can't answer that, but it's certainly, I can say in our plots that I have not observed a difference. Now, I also haven't sat down with a notebook and made pollinator counts per hour, but we get good fruit set on all of our Tetraploid Vaccinium ovatum. So that's I'm hedging my bet, but my anecdotal observation is that it hasn't seemed to negatively impact it in evergreen huckleberry, but I can see where it possibly could.
It seems like to me too that you're one part of this research breeding, and that it may be that another researcher might come in and examine things for effects on pollinators, like a team effort at some point. You're just one researcher, right?
Absolutely.
And I know that this disease resistance is also a small part of your overall research program, that you've released a few plants already. A couple of things while people are thinking of questions. One thing that jumped out at me was just the sheer amount of plants that you go through. And I was just kind of curious, how many plants do you think you would throw away, you said, typically? What's a percentage, just out of curiosity?
A percentage is 99%. But, so I think we have 60,000 plants in containers right now at the Lewis Brown farm. And, that's a little bit because probably 3,000 of those are cotoneasters that we're building numbers of to continue the testing. But yeah, you grow a lot and throw almost all of them away. So in our lilac program, I actually had those numbers not too long ago. Hibiscus is probably a more interesting one. So I think we grew 6,000 seedlings, 2014 and 15, and going into 2016, I think I kept 100. So after 2 years, I had thrown away 5,900 plants. Our burn pile is impressive.
Yeah, all your babies.
Yeah. Oh, it feels so good. When I was a graduate student, I didn't want to see anything get thrown away. Now it's like, it makes me so happy to go through and throw away plants because I have to pay for all of that space. It's very expensive.
Rhonda has a question. She's curious, what is the highest ploidy level in a plant?
Oh, so I should know that off the top of my head. Wait, did I just lose my?
Oh, I think you went out of presentation mode while we're.
Oh, okay. So, man, I should know that off the top of my head. I'm sorry. The largest genome size, I can tell you that the largest genome size is somewhere in the neighborhood of like 180 picograms. And so to put that in relation, Japanese cedar is massive. It has this giant pines, and Japanese cedar have these massive genome sizes, and they're about 16 picograms. So it's amazing how wide it varies. So plants can have as low as 2 chromosomes up to, we actually have developed 44X plants of Prunus laracerosus. So they have 44 sets, and they have 352 chromosomes. So it's really quite amazing. So sorry, I should know that, Rhonda. That's a great question. That's like a grad student defense question. I should really know that off the top of my head. I'm going to go find that answer and ask my grad student.
We can always put it in the notes later, right? Another question from Sandra. She's curious to know, and it may not be your research program, but if there's anyone doing research on reducing allergens in plants with a reduced pollen load. So there's a lot of beautiful plants that are popular, but a lot of people have pretty severe allergies. And so maybe there's a marketing, like a low allergy marketing angle.
It's a great question, and I had several ideas when I started working on conifers. So we've developed polyploids of occidentalis, platycladus orientalis, and Thuya plicata. And one of the things that I thought about was if we can increase the ploidy level of these plants, we might actually reduce the fertility and reduce the pollen load on those, because when we walk through the arborvitae rows, it seems like there's pounds of clouds of pollen. We have not noticed a big reduction so far in the amount of pollen that's produced in our polyploids. So it's not the main goal of our breeding program, but we're hoping that as we reduce pollen fertility, that it overall should produce less pollen. These triploids, they tend to have what look like normal functioning anthers, but then when you get up close, there's very little pollen on them. So I don't know if that sort of answers it. It's not our primary goal, but it is something that we keep an eye on.
We have two questions that are sort of somewhat related. So Zuann wants to know, how do you decide which plants to study? And then Rhonda's kind of curious, how many different plant experiments? So I know that you're not a plant pathologist, you're this overall ornamental breeder. So maybe just a kind of a quick overview of your program?
Yeah, so I'll answer the second one first. So we work on about 22 genera. And it's highly variable. Everything from, we tinker with penstemon a little bit. I don't consider myself a penstemon expert at all. And Galtonia candicans, Cape hyacinth, all the way, through most of our work is in woody plants. So even though I say we work on 22 genera, really most of our efforts are on 6 to 8, maybe 10, in any real, really big way. Probably most of our efforts are in maples, hibiscus, lilacs, and sarcococa maybe. So anyway, how do we figure out what we're going to work on? That is a great question. So I take a number of things into consideration. One is there a need from the nursery industry? So in Cotoneaster, there's maybe no real perceived need, but when I look at the ease of propagation, the ease of production, how well they do under drought and low input situations, that to me, they used to be grown by the millions in the Southeast, but now they're grown very little in the Southeast. And the main reason, two reasons I think, well, maybe three, but one is fire blight, one is phytophthora root rot, and the other is, this shift from, as Dr. Durr told me one time after going to the Home Depot to talk with one of their buyers, is everything's about color and green's not a color anymore. So yeah, it definitely has lost favor, but I think this or a paradigm shift to more low input plants will bring Cotoneaster back if there are better cultivars. So anyway, is there an industry need, right? Is the problem something that's within my ability to solve? So I had a pollinator scientist who came to me and said, you need to develop a tilia that is not attractive to bees. All right, well okay. So we could just shut down all flowering by developing a GMO, genetically engineered, using zinc finger nucleases and just shutting down all flowering in tilia, but that's not really within my skill set. It's not really feasible economically, and would it be adopted? Who knows? So feasibility, both within my skill set and within time frame, is a big consideration. And then there's also an interest factor. So is it something that I'm interested in? So not all the plants that I work on. So Philadelphus, for instance, is maybe not the biggest seller, and it's maybe a plant that it's kind of fallen out of favor. All plants, they have a 12 o'clock and a 6 o'clock, right? Everything goes in cycles. It's like fashion. Bell bottoms are going to come back. It's just a matter of time. Right now they're at 6 o'clock. And then skinny jeans are going to go out of fashion. Bell bottoms will come back again. So Philadelphus is going to come back in favor. But it's a plant. I love fragrant plants. And so we work on Philadelphus in a pretty significant way right now. Even though the industry wasn't knocking on my door, But I saw there's an opening and an opportunity to breed new, really good Philadelphus that have better fragrance than what's on the market. So again, I ramble a lot, but I hope that, there's a number of factors that go into it. So industry need, feasibility, economic impact, ecological impact, and my skill set and my interest.
Well, folks, I'm not seeing any other questions in the Q&A at this point. So, and I know we can't really clap because you're all silent, but we'll clap. And I really appreciate you coming, Ryan. Our next webinar is on not until March 12th at 11 a.m. And Nicole Sanchez is going to talk about identifying and managing the bronze birch borer. And look for an email coming out that also has a evaluation of this webinar. We'd love to get your feedback. So thanks so much.
Thanks a lot. I enjoyed it. Have a great day.
Thanks, folks. Bye-bye.